Okay, so in this video we are going to talk about the second part of the Gemini bar experiment. This is part B of the same handout, so you will just write one report. This week we are doing the heat treatment on the Gemini bar. So remember that you are testing 4 types of specimen, 1045, 1018, 4140 and 4340. And so the idea here is to compare the effect of alloying element on the hardenability of steels and also study the effect of carbon content on hardenability. So in this video we will talk more about the microscopic origins and the microstructures that relate to hardenability, and we'll shift to the harness testing that is performed in this experiment. So remember that in this experiment we are using a continuous cooling temperature diagram. They are called CCT diagram. This one is an example for the eutectoid alloy, so the 1080. And so what's important to remember is that we are looking at the temperature change as function of time for different cooling rates. So typically you will be starting in the austenite regime, and from this austenite we are going to quench the specimen at different cooling rates. So for instance, in water quenching we are typically in a cooling rate between 40 degrees C to 140 degrees C per second. And on the air quench hand, on the other side, we have slower cooling rates in the 5 degrees C per second. And so this diagram is useful because it gives you directly, if you know your cooling rates, the resulting microstructure. And for the problem of hardenability, we are interested in looking at the Martensitic transformation. So the Martensitic correction, remember, is what happens when you are quenching a phase center cubic unit cell. And which, if you go very slowly, will tend to form a body center cubic unit cell by diffusion of the carbon atom right here. But because we are quenching very quickly, under water quenching, this carbon atom doesn't have time to leave the unit cell, and as a result, we are forming a BCT cell, which is the structure of your martensite. Now what's special about this structure is it is no longer cubic, and therefore it will be very hard to deform. So this promotes the strengthening on the atomic scale that is contributing to the hardenability as function of heat treatment. So when we take a look at the 4340 alloy in particular, what we notice is the phase diagram is largely empty in the fast cooling rate. And so what this enables you is at fast cooling rate, we can produce a much larger volume fraction of martensite as opposed to the other alloy. And so if we look at here 4140 steel, which promotes relatively significant hardening, we see a direct correlation between the increase in hardness in the material and the percentage of martensite that is being formed during quenching. So in this figure, this represents the Rockwell hardness measured from the surface that is directly exposed to water in the Gemini experiment as a function of the distance from the surface. So this is here more the air cooled, and so here we have the water cooled heat treatment on this side. All right, so let's go back to our Lab4A experiment. So the TAs this week are going to post a video online that will show you an example of this Gemini experiment. And so it has two parts. One part will be water quenched very quickly, and the other side is simply air quenched. So throughout the length of the specimen, on which we're going to perform some hardness test here, you're going to achieve a gradient of microstructures. So these are examples of characteristic microstructures that you will see in a scanning electron microscope at a magnification of 500 times. Okay, so this will be your water quench end. And so what we notice here is that this microstructure is extremely uniform. And if we take a closer look, now we are at 1500 times, we see a formation of grains, but we see especially formation of needles of martensite. So on this picture in particular, you can see the martensite in those tiny sharp needles inside the grain. So if we take a closer look at 4500 times, we can see here the grain boundaries. We can see one grain here, there's another grain here, another grain here, and between them we see a grain boundary, which is very thin, so there's very little phase forming at the grain boundary, but we can clearly see the martensitic needles or lamella that are extremely sharp. And the faster the cooling rate, the sharper those needles. So on the other side, the air quench specimen for the 4140 steel, and it's going to show more of a inhomogeneous microstructure. If we take a closer look at 1500 times, it's made of two type of crystal. You have some plain crystal of ferrite. These are the BCC crystal. And we have some grains that are a little bit more distended. And so this represents your benite. It's also low temperature ferrite. Sorry, not ferrite, perlite. So low temperature perlite. So perlite should form some lamellar structure, but the benite here doesn't really form this structure. It's a lot more distended, but clearly different from a martensitic structure. Alright, so now let's take a look at the 4340. And so remember, 40 represent just the carbon content, so 0.4% carbon. And 43 represents the alloying element that have been added to the steel to contribute to the hardening. And so we can see here on the water quench end formation of large volume fraction of martensite, which is expected. But on the air quench side, for the other alloy, we will expect to see some benite. But here we see actually still a very strong concentration of martensite. There is still a bit of benite forming in the microstructure, but essentially you have martensite and benite. So clearly 4340 will show a relatively large hardening throughout the Gemini specimen. So let's talk now about the hardness testing itself. So in terms of hardness testing, we can divide the technique in two categories. And the first category is using an area-based approach, and that's based from the original definition of hardness from Brinell. So in Brinell, we press a hard sphere on the surface of the material, and we are measuring the size of the indentation, or more precisely the size of the projected area made on the indentation. So in this picture, that would be this distance here. And so this is a measure of the area of contact, which is used in the contact pressure the force supply divided by the contact area will give you a contact pressure that resists the penetration of the tip, and which is used here as a measure of a hardness. So in this context, the hardness represents stress or pressure that's representing the resistance of the material for penetration. Now, with Brinell, the only problem is when you use a relatively dull spherical tip like this, it requires you to apply very high loads. So a more practical test is using Vickers, which has a very sharp pyramidal tip. It's actually four-sided, like that shown here. And it is exactly the same principle. We are measuring the area of contact of this tip on the surface, usually done with an optical microscope. And from this, knowing the force applied to the tip, we are going to measure the contact pressure or the Vickers hardness. But here the loads are much smaller, typically less than 1 kilogram. And so it's much more appropriate for testing microstructures without damaging your component. So in your experiment, so before I move to the Rockwell harness, I want to say that the Vickers harness tester is actually very small in size. And so this is an instrument that you can really have on a table in your lab, very easy, about the size of an optical microscope. Okay, and so this will show, for instance, how you will convert your load in kilogram force and measure surface area in millimeter square to harness Vickers harness. Okay, but remember, this relates to a contact pressure. So this is another example of what you will see in the microscope, and the lines that are used to measure the two diagonals to establish the contact area. Alright, so let's talk about the Rockwell harness, which is the actual test performed in lab number 4B, not 6, just a typo here. Okay, 4B. So this technique is a little bit different from Vickers and Brinell. We apply the load, but instead of measuring contact pressure, this is measuring the penetration of the tip, the depth of penetration of the tip in the specimen. So in fact, you preload the specimen, so the tip will penetrate to a certain distance in your specimen, and so this is related to this gives you a constant E that will depend on the shape of your tip and the load you apply. Okay, and then you are going to remove that major load that's applied, but maintain a small load, so it's still in contact. And so what this Rockwell harness is actually measuring is going to be the rebound distance from the reference line at here. Okay, so basically it's some sort of measure of elastic rebound. The harder the material, the larger that rebound in the specimen, and the material that's more plastically deformed by indentation will show very little rebound. That means it's mostly plastically deformed. Okay, so what we're measuring is really this number here. So if we compare this approach to Vickers and Brinell, there is a distinction between these methods. In the case of Vickers, we again measure stress or contact pressure, and you can show this is actually the Vickers harness is 3 times the tensile stress in the material. So it's measuring a stress. As opposed to Rockwell harness, which takes into account of the elastic recovery and will measure the permanent deformation or strain in the specimen after unloading. So Rockwell measure or relates to permanent strain, and Vickers relates to the yield stress. So what is going on during the test? In general, this constant E is fixed. It depends again on the type of Rockwell tester you are using. And so it will be predefined. And it will measure the depth of penetration or the difference in penetration from the minor loads you applied and the major load. Okay? Now, an important factor in this test is going to be the time that you allow the specimen to relax during the application of the load. So if this time is too short, you may not achieve maximum penetration. So there needs to be a minimum dwell time when you impose the load. Okay? And so this test, unfortunately, because of this dwell time, is relatively time consuming. So typically in the experiment, we will take some time to produce harness along the length of the Gemini bar. So this week the TAs are going to conduct this test, and next week we will post a video more focused on the Rockwell harness testing, so you have an idea of the practical aspects of the test. Alright, so to summarize the Rockwell harness tester, we have an application of a minor load followed by a major load and then noting the depth of penetration. So that's the principle. The harness value is directly measured from a dial, in which a harder material gives a higher number, that is a higher rebound, a higher elastic rebound. So the advantage is the harness values is directly displayed. There's no calculation like in Vickers, and there's no optical or physical measurement in this technique. So in that sense, maybe the analysis is a bit more simple and faster. And so in the laboratory we follow an ISTM standard E18, which describe in detail the methodology for Rockwell harness testing. As I said, we have different type of Rockwell harness. So typically you would put your harness measurement with a letter, so such as HRA, HRB, HRC, to indicate the type of Rockwell instrument you've been using. And so the difference between the Rockwell tip basically will be maybe the shape or the material of the tip that's being used. It could be spherical, conical, so more or less sharp and made of diamond or steel. And these are examples of different tip that can be used with Rockwell harness testing. Alright, so this will depend on your application and the amount of material you have available. And of course, to make sure we get proper measurement, the machine is calibrated with reference specimen. So of course, one inconvenience with this Rockwell technique is it produces a relatively large uncertainty. There are different factors that influence the measurement. For instance, the standards that are used, the type of material you are testing. So some Rockwell harness tip will not work well with certain material. Since it's measuring directly the penetration in the specimen, the thickness of the specimen is going to play a role. If your specimen is not perfectly flat, in particular, or is tilted, this will influence the penetration. So a lot of uncertainty with Rockwell that may not appear with the other two techniques, Vickers, because they are only measuring a force or a pressure applied to the specimen. The width of the area to be tested is important. So if, for instance, if your harness test is too close from the surface, this will influence the actual depth of penetration. The dwell times are important, and since we are performing Gemini experiment where the specimen are exposed at high temperature, you're gonna see on the specimen an oxide forming, and so it's very important to remove this oxide or you will affect or influence the depth of penetration of the specimen, which also could be a problem if your specimen is too thin, because you are removing a certain amount of material due to the oxidation. All right. And finally, something that's maybe important to understand is there's always a way to take one technique and convert this measurement with the other methods. But in general, we're going to use certain technique like Rockwell, Vickers, or Brinell, depending on the type of material we're using. So in general, personally, I like Vickers because it's applicable to quite a lot of materials, most material in general, even very hard ones like ceramics. And Rockwell is a little bit more sensitive to the type of material. So you'll have to adjust the type of Rockwell, the tip and the load you apply, function of the material. Okay. So it requires a little bit of expertise when you use Rockwell or understanding of the material you are testing. Alright, so this concludes this week's video. Again, our TAs are going to post online videos for the key part of this experiment, and we're gonna post the data for the 4 specimens that have been should have been tested this week before we went online here. I wanted also to clarify some question I had about lab reports. So this week we have a lab report number 3 that will be due on Friday. So the TAs are going to reset the deadline for this on Blackboard. We are only communicating on Blackboard for the lab reporting. Lab number 4 will be due, if I look at my schedule, a little bit later on the first week of April. Okay, so there's gonna be next week a break for lab report, and then the next report will be due in 2 weeks from Friday. All right, and I will post a new video for the next experiment, number 5. All right, thank you very much.